Hey, good morning, everybody. We're just going to give it another couple of seconds just for the last people joining, and then we will start this webinar. Okay, perfect. So as the people are joining, I'm going to get started, Salang. So welcome, everyone, and thank you so much for joining us for this webinar this morning. My name is Monique. I'm the application specialist responsible for IDT at Whitehead Scientific. And with me today, I have the rest of the team. So we have Megan, who is our product manager, and we have Tynan and Letitia, who are our sales specialists, also responsible for IDT. Most of you may know their faces already. And then, of course, we also have Sipalele, who is part of our team. She manages our Oligos mailbox. So if you send anything to our Oligos mailbox, she will probably be the one to respond to you. And if at any point you have any questions regarding today's webinar or anything IDT related, you are most welcome to reach out to us. I will also share our contact details after this webinar. And with that, I'm going to start sharing my screen. Okay, I believe you should be seeing my screen at the moment.
Yes, thanks.
So I'm going to, okay, perfect, thank you. Okay, so I'm going to jump right in. So the webinar today is going to be focused on IDT, and I'm just going to provide you all with a very high level overview of the products that we can offer from IDT, and I'm going to show you where to find some information on the IDT portal itself. I'll also do a quick run through of the free online tools available that we have. And then we are also launching our own how-to page. And we will share the details of that website with you after the webinar, where you can access any frequently asked questions. We have some videos to show you how to navigate the portal and some extra information for IDT, which will help you on your journey in terms of custom DNA synthesis with IDT. So let's get going. Who is IDT? Well, IDT is a genomics company. IDT stands for Integrated DNA Technologies, for those of you who are not aware. And they started out as a oligo manufacturer, and they've really grown into one of the leading genomics providers in the world. And they're a very exciting company to work with. They currently employ more than 325 engineers and PhD students who work behind the scenes, or PhD scientists, sorry, who work behind the scenes to do product development. They have advanced technical support on all of their products, and they really expand their product offering to serve the market in terms of genomics research. At the moment, they have more than 120,000 customer publications. Again, just highlighting the fact that IDT really provides high quality products, enabling genomics researchers across the world. They supply their products in more than 100 countries, and every year they serve more than 130,000 researchers in the genomics space. So if we look at the product offering from IDT, it can be broadly categorized in the following groups. So of course, we have our oligos, and these can be DNA and RNA products. We have standard oligos, we have oligos with modifications. So any application where you would need an oligo, we can probably help you with that. And I'm going to provide you with some more information in terms of the various options we can offer in terms of oligos in a couple of slides. Then we have our CRISPR gene editing portfolio, where we have a variety of Cas enzymes to choose from and some tools that can be used for your CRISPR editing experiments. We can do gene fragments and gene synthesis. We have quite a big variety of next generation sequencing products. And then, of course, our PCR, qPCR, digital PCR offering, which is quite popular. Now, in the next couple of slides, I will just highlight the main products under each of these categories. So the first category I'm going to look at is our CRISPR gene offering. So under our CRISPR solutions, we have our CAS system. So at the moment, we can supply Cas9 and Cas12a, depending on your need in the lab. We can do guide RNA. Under guide RNA, we can offer single guide RNA, so where your CRISPR RNA and your tracer RNA has already been complexed into a single molecule, or we can offer them as two separate molecules, a tracer and a CRISPR RNA separately. So whatever you may need in the lab, we can offer in any configuration that you may want. And we also have some modified Cas enzymes. So if you're interested in an example for looking for a Cas enzyme that has been fluorescently labeled to look at gene editing, we can help you with that. We also have some nicases available. So if you're interested in modified Cas enzymes, please do reach out to us and we can send some more information your way. Then we also have a guide RNA selection and design tool. So if you are, for example, looking through academic literature and you find some guide RNA that may be applicable to your target that you're interested in, you can check those guide RNAs on the IDT portal. And this is free to use. You just log in with your IDT portal details, go to the tool and you plug in the information. And the tool will then give you some on-target and off-target scores depending on the organism that you are working with. And then we also have some pre-designed guide RNAs. So if you are working in human cell lines, mouse or rat samples, we have some pre-designed guide RNA. So pre-designed assays for specific genes. And you can also use this selection tool to browse those pre-designed assays. They come again with on-target and off-target scores that you can look at, and then you can very easily add them to your cart and order them if those are applicable to you. Then, of course, very importantly, when you do CRISPR gene editing, you want to make sure that your gene editing has actually taken place and that it has the desired effect. And to do that, you want to check your cells that you have edited. And we have two options in this space. The first one is a PCR-based kit that you can use to look at your genome editing. And this is our Alt-R genome editing detection kit. So you will run the PCR assay as described in the instructions for use, and you will then run a gel and analyze the bands on the gel to determine whether your editing was successful. Now, this is not necessarily the most sensitive technique. It just gives you a look and see to see if your editing has been successful. So if you want more information, we do also have a next generation sequencing offering, which is our RAMP-seq CRISPR analysis system. We also have analysis software that goes with this kit. So it's a next generation sequencing workflow with an analysis tool that is available online. Our next category is our gene synthesis and fragments category. So under genes and gene fragments, we also have a variety of products, and it really determines on what you plan to do, or it really depends on what you plan to do in the lab, and that will determine which product we will offer you. So the first product offering is our G-blocks and our G-block hi-fi fragments. So these are double-stranded DNA fragments, and they are ideal if you want to do some cloning experiments or if you want a positive control for qPCR assay, for example. They can go up to 3,000 base pairs, and then depending on the size of your fragment, you will get a certain amount delivered in a tube. Then we also have E-blocks. Our E-blocks are very similar to our G-blocks in that they are also double-stranded DNA fragments. However, we supply them in smaller amounts for quicker turnaround time, and they only go up to 1,500 base pairs. The E-blocks are ideal if you're looking for a more cost-effective solution, but if you're also looking for a higher throughput screening solution. So we generally offer these in plates. So if you want to screen a variety of gene fragments for different mutations, for example, this would be the product offering we would look at for you. Then we have custom genes. So if you have a gene of interest that is larger than what we can offer with a G-block, we can look at custom genes that go up to 5,000 base pairs. And these genes are also double-stranded, but they are supplied in a vector. So these come within a plasmid, and at the moment, IDT has the following vectors available. So you can order your gene fragments in any of these plasmids that may work for you. You can use these fragments as a positive control in your qPCR assays, for example, but you can also use them to subclone into your vector of choice, or to just do cloning experiments, or to do some bacterial mammalian expression. And these are the vectors that you can then choose from. So at the moment, IDT does not offer a custom vector synthesis solution. So these are the only vector backbones that we have available. And you can find some more information on the IDT portal as well. So if you wanted to look at the gene sequences of these vectors, as you can see on the right-hand side here, we have the sequence of these vectors in a text file, a gene bank file, or a PDF. So you can download it and check it before you order any of these gene fragments. Then very exciting, IDT has launched their vector onboarding service. So if, for example, you have a vector that you are using in the lab quite often, and you want to clone multiple gene fragments into that vector for certain experiments, you can now onboard your vector with IDT. Now, with this service, what you would do is go onto the IDT portal, log into your account, and then go onto the vector onboarding tool. The tool will then ask you a variety of questions. You will name your vector, you will give us the sequence information, you will tell us whether this vector can be cloned into E. coli, whether it has selection markers, and then you also tell the tool where in that vector your insert needs to go. So you will give it some gene sequences before and after the cloning site, and you will upload the gene or the vector backbone. Now, for the vector onboarding, there will be a cost involved, so once-off cost to onboard the vector. And once you've gone through all the steps in this tool, we will be able to generate a quote for you in terms of what it would cost to onboard that vector with IDT. Once you have then generated your PO for the onboarding and we've processed that, we organize together with IDT and your lab to then send that vector to IDT. So very importantly, this needs to be a vector that you have in the lab already. So in a tube available so that we can ship it to IDT. IDT then keeps that vector in storage. They do some QC as soon as they receive it, and they then keep that vector on your profile. They load it onto your profile, and any genes that you would want to order, you can then order in your vector of choice, in the vector that you've onboarded with them. Also, very importantly, IDT does not synthesize the vector. So it's not a case of providing the sequence and they synthesize it. You physically need to ship your vector to IDT, and they also do not make changes to that vector. So you need to make sure your backbone is exactly what you want it to be. But if you are looking for a lot of clones generated in your vector, this may be a very, very easy way to generate a lot of vectors with your different genes of choice in that vector. And IDT then does some QC in terms of next generation sequencing and sends you that vector back ready to use in your lab. If you need any more information on the service, please do reach out to us. We would be happy to discuss this further with you. And then our last product offering is our single-stranded DNA. So if you need fragments in the range of 200 to 2,000 base pairs, and you need single fragments because, for example, you are doing homology-directed repair, then our megamer single-stranded DNA fragments will be the product of choice. For the megamers, you can order your sense and antisense strands. So whatever you may need in the lab, we can probably offer that product for you. Okay, our next product offering is our next generation sequencing portfolio. So under our next generation sequencing solutions, we of course have library preparation kits. Now these kits have been optimized for sequencing on Illumina. We have library preparation kits for DNA samples. We have mechanical as well as enzymatic fragmentation options. We have kits available for low input samples, as well as dedicated kits for FFPE samples. So if you are looking for an alternative kit to use with your Illumina sequencer for DNA sequencing, please do reach out to us. We will have a variety of options that we can discuss with you depending on the sample type that you are working with. Then we also have some RNA sequencing library preparation kits, also for Illumina sequencing. And then we also have methylation sequencing kits available. So if you are looking for Illumina-specific sequencing kits, please do reach out to us in terms of our library preparation offering. Then if you're interested in targeted sequencing, we can also offer you a variety of options. So one of our offerings is our hybridization capture panels. Under hybridization capture, the capture reagents, we have our core reagents that you can order from us, and then you can do custom or already designed panels. So at the moment, hybridization capture is ideal if you are looking at larger targets or larger panels. So what you would do is you would do library preparation of your samples beforehand with your kit of choice, and that can be with our IDT kits, which is recommended because we can help you troubleshoot that. But you can also use an Illumina library preparation kit. We have blockers for some PacBio and MGI systems as well. So if you're using a different sequencer, please also reach out to us in terms of our targeted sequencing options. We will probably be able to help you with that as well. Then once you've done your library preparation, whether with the IDT kits or another library preparation kit, you will then capture your targets with probes, extract those targets by binding those probes to streptavidin beads, clean it up, and then sequence on your sequencer of choice. At the moment, as I mentioned, we have custom panels. So if you have a panel that you've seen in literature or you've designed your own panel, we can do a custom configuration for you. But we also have pre-designed panels like our whole exome sequencing panel, which has been performing really well in the market. Okay. Then, if you would rather do amplicon sequencing, we do have amplicon sequencing solutions as well. Our amplicon sequencing panels are ideal if you want to look at smaller panels, smaller gene targets, then amplicon sequencing is ideal. Under amplicon sequencing, we have panels for cancer research, metagenomics, microbial sequencing. Very popular is the SARS-CoV-2 sequencing, where you receive all the reagents to do your amplicon generation, and then that can be coupled with your library preparation kit of choice downstream, ideally using the IDT library preparation kits. Under amplicon sequencing, we can also do custom panels. So if you have a panel that you have designed or that you've seen in literature, we can also generate the primers for custom panels for amplicon sequencing as well. Then we have our Archer IDT offering. So Archer looks at genomic variation. It's a very sensitive kit. So if you're interested in our Archer offering, please do reach out to us. Then we have a library normalization product line, which is our Normalase module. The Normalase module can be used to normalize your library so that you get equal coverage for your different barcodes within a library. The Normalase module is compatible with most of our library preparation kits and can also be used in combination with hybridization capture or amplicon sequencing. And it allows you to very easily achieve equal coverage over different barcodes in your final library. It is a PCR-based kit, so you will perform a Normalase PCR to balance your barcodes instead of having to manually do it by calculating the amount of library per barcode before combining them. So a much, much easier way and a much more efficient way to get equal coverage across your barcodes. And then, very importantly, we can also offer sequencing adapters and primers for a variety of sequencing platforms in a variety of configurations. So on the IDT portal, under our adapters and primers section, you will see this page pop up. So we can do adapters and primers for Illumina sequencing, for Ultima Genomics, Element Biosciences, and MGI. And we also have a custom sequence adapter configuration tool that you can use if you have custom sequences that you would like to order for your next generation sequencing experiments. And if you have no idea which option to choose, you're more than welcome to reach out to us and we can help you navigate the different options as well. Then probably one of our most popular categories is our PCR, qPCR, and digital PCR offering. So this is mainly focused on our probes that are available. It falls within our oligos category, but we've kept it separate just because there are some iterations under probes, which sets them apart from just modified oligos. So with our IDT PCR, qPCR, and digital PCR products, our probes are all HPLC purified. We can also offer for longer probes page purification or any other purification that you might be interested in that's specific to your application. Our probes have a very high efficiency and they come with very stringent quality control parameters. So you know that when you order a probe from us, you will get a very high quality product. We currently have more than 18 fluorophore and quencher combinations available. And this can be configured on the IDT portal, but you're also welcome to ask us if you're not sure which combinations to choose. We offer various packaging sizes, so various scales of synthesis to suit your budgeting needs and also your experimental needs. Then unique to IDT is our double quench probes. So as a standard, probes come with a fluorophore on the 5' end and then a quencher on the 3' end. Now what IDT has done is they've developed a product where a second quencher is integrated into the probe and it usually sits in the middle of the probe. And this double quenching on the probe has actually improved sensitivity, so you can detect your target at a lower CT value during your qPCR assay, and it also reduces background noise. So if you are running qPCR, we would definitely recommend trying our double quench probes to see if it improves your experiments. I can guarantee that you will definitely see lower background and you will definitely see improved sensitivity. Then we can also offer locked nucleic acids, molecular beacons, and we have some pre-designed assays. So on the right-hand side of the screen, you will see we have microbial identification assays. So these have been pre-designed. It's a qPCR probe set. And you can see the catalog numbers on the right-hand side there. So at the moment, we have already designed probe sets for Clostridium difficile, HSV, influenza and RSV, MRSA, and Candida auris, amongst others. So if you are interested in any of these organisms, also please do reach out to us. And then probably one of our biggest categories is our oligos offering. So this includes our PCR and qPCR primers and probes, but also oligos for any other application. So we can do custom DNA and RNA oligos with or without modifications. At the moment, IDT offers more than 1,000 modifications for oligos. These include 5' and 3' modifications, as well as internal modifications. And all of these modifications are available online. If there's a modification that you are interested in that you can't find online, please do reach out to us, because IDT also have non-catalog items, which is not on the website, but that they can still offer you depending on your experimental needs. Our DNA and RNA oligos are also offered in various packaging sizes, so various scales of synthesis to suit not only your experimental needs, but also your budget. We have various purification methods to choose from, whether that is standard desalting, HPLC, or page purification, and some other options as well. And then we can offer a variety of lengths in terms of your DNA and RNA oligos, depending on what your application is. We can also do single-stranded DNA, double-stranded DNA. We can do pools of oligos, whether that's primer pools or pools of other oligos that you want to combine before ordering them. And we can also do aliquots and replicates. So if you need any oligo in any sort of configuration, please do reach out to us and we will be able to assist you. Now, you may be wondering, this is a lot of information. Where do I actually go to get this information? So the first point of call is our IDT portal. So if you type in this web address or if you Google IDT DNA, you will land on this portal, and this is what it would look like on the computer screen here. And I'm going to show you a video now of how to navigate the portal and where to find some of this information. So this is what the portal would look like once you have Googled it. This is the landing page. Now, at the top, you will see some, I'm just going to grab my pointer here. So at the top, you will see we have these tiles, we have products and services, Solutions, Education, Tools, and Company. Now, these are drop-down menus for easy navigation online. You will also see at the top here a sign-in button where you can sign into your IDT profile, and this is very useful if you have a profile to access all the tools and to generate your own quotes. At the top here, you have your IDT button, which will always bring you back to this landing page if you have navigated the portal and looking for information. Okay, so I'm just going to start this video. Okay, so if you scroll down on the IDT portal, you will see this landing page that I've shown you this whole morning with all the categories of products. And you can then click on those tiles to look at all the different information. So it will pop up with more tiles in terms of the products available on it. And you will see that it also has a description of each of the categories. So under our oligos, we can see here our custom DNA products and also our modifications. Then if you scroll down, there's some more information on the synthesis of IDT oligos, and we also have our resources listed. So if there is some information that you're looking for or frequently asked questions, you can just scroll down. You can also navigate to these pages using the drop-down menu at the top, which will bring you back to this page, and you can go into the different products available. Now, if you click on these tiles, it will take you to another menu where you can get more information on all the different products. So under our custom DNA oligos, we now see all our options. So we have DNA oligos, ultramers, our oligo pools. We can even do mix-based oligos and affinity plus or locked nucleic acid oligos. Again, if you scroll down on this page, you will see some more information in terms of the products available and also modifications, the synthesis process, as well as our resources available. Okay, so that is how you can navigate the IDT portal. So very user-friendly. You click on the tiles and it will take you to more information and to more of the products. Now, one of the questions we do get quite often is in terms of modified oligos and where do I find information in terms of modifications available from IDT. Now, very, very easily, we are going to navigate the IDT portal again. The video will play in a second. Okay, so we can scroll down to our oligos category once we've logged into the website. Click on oligos, and under oligos, you will see again our product offering, and we can then click on oligo modifications. The oligo modifications will then take you to a menu where there are all our different modifications available. Of course, fluorophores and quenchers are our most popular in terms of qPCR. And then we have some other modifications as well available, like our attachment chemistry and linkers. So biotinylation, for example, we can do phosphorylation. We have carbon spaces, click chemistry modifications, phosphothiorate bonds, and modified bases available. And we have a variety of options under each of these categories. And if you don't find your modification under these categories, you can reach out to us and we can check if there are non-catalog items available. If you scroll down on this page, you will also see this explanation of the different categories in terms of modifications. So what each category represents, and then also examples of the modifications we can offer, and then applications that these modifications are usually used for. And right at the bottom, again, our resources available that you can click on and use as needed. Now, if we scroll back to the top of this page and we click on one of the categories, it will take us to another menu where you will see a more finer categorization of the different modifications. In this case, we're looking at our fluorophores and dark quenchers. So we have Freedom dyes, which has no patent licensing restrictions that IDT can offer. And if you click on Freedom dyes, you will see all the dyes available. It will also give you a description at the top of what you are looking at. and then a list of modifications available. Very, very nice on the IDT portal. It also tells you where these modifications can be used on your oligo. So whether that's 5', 3', or internal, it gives you the excitation and emission wavelengths, and it also gives you alternative dyes if you are interested in an alternative listed. And then again, at the bottom of the page, we have our frequently asked questions and our resources. The same thing for additional fluorophores. Here you will see it summarized in a table, again showing you exactly where the modifications can be used, the excitation and emission spectra, alternatives available from IDT. And if you hover over the different options, you will also see some pricing information on the portal before you have even generated a quote. So very, very useful page to use. Then, of course, with our fluorophores, we have our quenches available. If you click on the quenches, you will find a menu like this where it will tell you the product and then again where those quenches can be used in your sequence. We also have other dark quencher modifications. And if you click on these gray tiles available, it will take you to some more information in terms of the structure of your dark quenchers if you're not sure which dark quencher to choose. Now, apart from fluorophores and quenches, we also have some other modifications like our attachment chemistry and linker modifications. As you can see, it shows you exactly where the modifications can be used in a sequence, and these are all the listed ones currently available on IDT. But as I mentioned, if you're looking for a modification that's not listed here, please do let us know. And then if you click on those gray tiles below each product, you will see some molecular weight information, the modification code, as well as the chemical structure. So you can double check whether the modification is actually what you are looking for. Okay, so most of the IDT web portal can be navigated like that, just clicking on the different tiles, looking for the information. Now, one of the other most important things that I want to show you today is once you have all the information, once you know what you want to add on the portal, how do you actually go about generating a quote? So also very, very easy. And this video will also be available after the webinar if you want to go look at it again. So on the IDT portal, you will go to the IDT website, which I've shown you. You're then going to log into your IDT account. If you don't have an account yet, you can just click on register and we'll take you through the steps. Otherwise, you can sign into your account. And this will then take you to a landing page, which will look like this. And say, for example, you want custom DNA oligos for this quote. It will then take you to the oligo entry tool. Here you will then select how many items you want, and you just populate all the information that you want for your specific product. So your sequence information, unique names for each sequence, the scale of synthesis, purification, and once selected and added together, you say add to order. Very importantly, when generating a quote, follow the orange buttons on the portal. Once you've added to cart, you can check your cart, make sure that you are happy with the products you have added, and then follow the orange button to check out. Under the checkout option, if this is the first time you're generating a quote, we will ask you for your shipping and billing information. This will only be done the first time, which you can then submit. And then you will see this page where you have submit your order and request a quote. You then select request a quote and submit. And the portal will send that quote directly to your email address. And there you have a quote. Super simple, super easy, takes you about a minute to generate a quote. And you can then use that quote. It is an official quote that you can use to generate a PO on your side. One of the other questions we do get quite often is help in terms of primer design or assay design. And so I just want to show you this video to show you the IDT tools that are available. So if you have an IDT portal login details, you can log in and use any of these tools free of charge as many times as you want. So again, we have our IDT portal page where we come in and you can then log in to the portal page. And at the top, we have our tiles, which show our drop-down menus. And if you go to tools, you will see there is a variety of tools available from our Oligo design and handling, qPCR assay design to our NGS tools with a variety of options available. So most of the products I've touched on today, there is a tool available that you can use either to configure your oligos or your product that you want, or to help you with a design. So one of the tools that I want to show you today is our Oligo Analyzer tool. So if you want to do primer design or probe design, you can use our Oligo Analyzer tool to check your design. So if you already have a sequence or you found a sequence in a paper, you can go to this tool online. This is what it looks like, and it gives you some pointers. If you're not logged in, it will ask you again to log in. And once you are logged in, it will take you to a page where it will ask you to input your sequence information. So this is what the page would look like. So you just paste your sequence information in the white block where it says sequence, and then you also have the option of adding modifications on the 5', 3', internally or mixed bases. On the right-hand side, you then set your certain parameters for your PCR assay that you're going to run, and you then click on Analyze, and it will give you a report of any secondary structures that your oligos may form and give you a score in terms of how good that oligo has been designed. Once you're happy with your design, you can then obviously order it. Now, once you've ordered oligos from us, you will get it lyophilized. Now, a lot of you may wonder, how do I resuspend my oligo? Well, very easily, you can go to the resuspension calculator. On here, you can add the quantity you've received, change the values, and also the units that it is given in. And you can then click calculate, and this tool will then tell you exactly how much water or TE buffer to use to resuspend your oligo. We also have a dilution calculator, so you can tell us what your starting concentration is and what your volume is and how much you want at the end. You can also navigate the different amounts or concentration units that is available. And once you've populated all the fields, you can click calculate, and it will tell you exactly how much of your stock you need to dilute and how much water or buffer to get to your desired concentration. Then if you are designing a new primer assay, whether that's for PCR or qPCR, you can use our Primer Quest tool. The Primer Quest tool looks like this, where you can insert parameters as well as your sequence information, and it will give you primer and probe options for that specific assay. So you have a variety of options here. You can enter your sequence manually, you can enter the NCBI ID number, or you can upload Excel files. At the bottom, you then choose whether it's a PCR or qPCR assay, and it will then, based on the sequence information provided, show you custom design parameters with primer and probe options, which you can then directly add to your cart and order as well. For our CRISPR genome editing options, we have the Cas9 selection tool, design tools. You can check your HDR designs as well. For qPCR, we have a variety of tools as well. Again, the Primer Quest tool that we've just looked at, and very useful, our multiplex dye selection tool as well. So if you're using an instrument and you're not sure which fluorophores you can use with that instrument, this tool can help you navigate that. And I do apologize, this is going to load for a little bit, so I'm going to just fast forward this this video quickly. There we go. Okay, so the primetime multiplex dye selection tool then allows you to select your qPCR instrument of choice. You then add how many assays you can do. Mostly it will be by default, but if you have less assays, you can change the number of assays. And at the bottom, it will then give you dye options and combinations that you can use. And it will also list alternatives. Once you've configured your dye options, you can go to next and then add your primer and probe sequences and configure your primers and probes for your specific instrument. For all our other categories, we also have some other tools that you are welcome to scroll through on the IDT web portal. I'm not going to go through all of them at the moment. The last one I just want to show you is our adapter configuration tool. So if you are doing next generation sequencing and you want custom adapters, this is our custom adapter configuration tool. You will go through all the steps where it will ask you certain questions that you can go through. And at the end, it will give you a product configuration, which you can then directly add to cart to generate a quote or to place your order directly on the portal. And I know this has been a mouthful this morning. So to make your life even easier, where you can learn where can you find some more information or learn more about the IDT portal? We are launching our IDT How-To page that is hosted by Whitehead Scientific, and the details for this will be shared with you after the webinar. On this webpage, you will have various information sources. We have how-to videos. We have frequently asked questions. So if you do not know where to start, this webpage will be a great place to start where you can get more information. Our contact information will be there. So please do make use of this and reach out to us if you have any questions. And with that, that is the end of my presentation. I would be happy to take some questions if there are any. You're also very welcome to type your questions in the chat if that's easier, or in the Q&A box.
Melita, so the recording together with or an email together with the recording will be sent out to all the participants that registered for the webinar. So you should be able to get that a little bit later today. Monique, there's a question saying, thanks for the talk. Could you go over your codon optimization tool?
Sure. Let's see if I can log in quickly. I can share my screen. It is loading on my side. I will share in a second. Okay, just going to stop sharing the presentation quickly, and then I will share my computer screen on the side. Okay.
Can see your screen. Thank you.
Okay, perfect. Because my Teams just went dark, so I wasn't sure if it's still loading. Okay, perfect. So this is the codon optimization tool once you have logged into it. So very nice. You have these info buttons that can give you more information in terms of what the tool does. Then over here at the top, the first thing that you need to populate is your sequence type that you are using, whether that's DNA bases or amino acids. Then the product type that you are interested in. For DNA bases, we have our E blocks, G blocks, our genes, and our megamer option. If we were looking at amino acids, same options available. So you just toggle between the two, select your product that you are working with, and then you choose your organism that you are working with. So you can see there's quite a list of organisms here. Choose the one applicable to you. And then you can do a single or a bulk entry. Bulk entry is just like a FASTA file format or an Excel sheet. You can also navigate between the different files that you can upload. Single entry, you just paste the sequence here. And then you can use the buttons on the side here to highlight your codons, map the codons, and to optimize based on the organism that you are working with. If you have restriction sites, we have information on that as well available here. The same with the bulk entry tool. You can map your codons and optimize them. And that is it. So the tool does everything else for you once you've populated it. We also have at the top here, I don't know if you can see the blue links here, but there's a decoded article on how the tool works. So the more finer workings behind the tool. And we also have a video to show you exactly how to use this tool and interpret the results that you get from this tool on here. So I would definitely encourage you to go watch these videos and read the article as well. And if you get stuck at any point, you're welcome to reach out to us. So I hope that gives you an idea of what the tool can do.
Thanks, Monique. Also have another question. Do you have tools to assay microRNAs, 18 to 22 nucleotides?
So we do have, let me just double check while I'm on the portal here. At the moment, there is a DSI RNA tool, and there is also, sorry, it's just loading quickly. RNA interference design tool, but there's no specific myRNA tool. I do believe if you need help, you can definitely reach out to us. And there should be technical support from IDT side as well to help you with your myRNA designs as well. So we can definitely look into that for you and get back to you.
Thanks, Monique. Are there any other questions? I'm going to check in the Q&A. Let's see any there. Q&A. Just give it 2 more minutes just to see if anyone else is still typing.
While we're waiting, I will share our contact details on the screen. So if anyone has any other questions and would like to reach out to us, then you have that as well. So I'm just going to share my screen again. Go to the next slide.
Okay, I don't see any other questions coming up. Thank you very much, Monique. And thank you for to everyone who has attended. And if you have any questions, please feel free to reach out to us.
Thank you, everyone.
Thank you. Bye.